Okay, so let's get started with our seminar series for the fall. We have today Dr. Jennifer Sang, who is an assistant professor in the Department of Biochemistry and Microbiology here at Rutgers. And Dr. Sang is an accomplished molecular biologist and entomologist whose research explores how insect-associated microbes, especially those outside the gut, influence insect sensory systems and behavior, with implications for disease transmission and vector control. Her interdisciplinary work bridges microbiology, neurobiology, and ecology, with recent discoveries demonstrating how common microbes like E. coli can directly reprogram insect behavior. Dr. Sang has training at Yale and Princeton, holds multiple prestigious fellowships and awards, and has authored numerous impactful publications spanning topics from olfactory physiology in Drosophila to current sensing in bacteriophage host interactions. She's also a dedicated mentor, educator, and leader within the scientific community. So please join me in welcoming Dr. Jennifer Sang.
I'll cook this and just check what you're all seeing. Okay. All right. Now how do I click on the Okay. I don't think that worked. Yeah, let me click this arrow. Okay, perfect. So yes, thank you for inviting me to speak about my research, which studies the insect microbiome to advise the development of sustainable solutions for global health and environmental challenges. So as you're probably aware, insects present a huge burden on the human population as they can cause significant agricultural damage, including to our vegetables and our grains, as well as spread diseases like malaria and sleeping sickness. Now, these insects utilize their sense of smell to detect their mates, insect repellents, food sources, and even you and I. So a better understanding of the olfactory system, particularly the factors that regulate its function, could lead to the development of alternative strategies for insect control. Okay. So on my team, we hypothesized that the composition and diversity of the insect's microbiome modulates their olfactory receptor expression, as well as downstream neural processing, thereby influencing the host insect behaviors, including foraging, mating, and habitat selection. So one arm of our research is to investigate those insect-associated microbes and their roles in modulating insect olfactory behavior. Considering the fact that microbes are, at least in part, horizontally transmitted by plants and water sources to their insect hosts, we further hypothesized that environmental factors, including longitude, latitude, temperature, and precipitation, can all impact the abundance of these symbionts and the microbial diversity within the insect. So that analysis of the microbial community structure can enable the identification of drivers for diversity as well as indicator strains for specific conditions. And our investigation of the interactions between these microbial communities, whether it's in plants, water, as well as in the insect, these will all advise the use of microbiome exchange for everything from crop protection to insect control. Now, indeed, other groups have conducted microbiome exchange studies, which demonstrate that there is an overlap of key microbes between these plants and the insects. And the interactions between each species of plant, each species of insect, and their microbiomes, this is likely highly specific and dependent on the feeding mode and environmental conditions of the insect. Now, we were particularly interested in seeing if this microbiome exchange can occur in chemosensory tissues, where the microbes could have an influence on olfaction. Specifically looking at the labellum, which is their mouth part, and then the tarsi, which are their feet. Now, something I did not mention earlier is that most of the research into these insect-associated microbes has been focused on the gut microbiome. But microbes are certainly present throughout the insect, such that they could influence olfaction in those non-gut tissues. So we surface sterilized, then we collected the mouth part and the feet of laboratory strains of mosquitoes. These are the regions of the mosquito that are most enriched in the chemosensory receptors. And then the resulting sequencing data, we quality checked it, aligned it to the mosquito's genome. Any reads that were not mapped to the mosquito's genome were then used for metagenomic analysis to identify all of the fungi and the bacteria that are present. So if we find anything here in our laboratory strains, we would expect to find microbes in our field samples. And from that analysis, we did discover an extensive profile of bacterial families that are represented in the foot and in the mouth parts. And moreover, the abundance of select families differed after the mosquito took a blood meal, and we see this in the foot as well as in the mouth part. So perhaps this is evidence of resource competition. This is also evidence that microbes are not just inherited, but they're being acquired from their environment. And that acquisition is not limited to when they're young, but it can also happen later on when they are adults. And it's fluid with the environment that they're encountering. And more importantly, this is evidence that microbes, most likely facultative symbionts, they are present in these non-gut tissues. So there's a possibility that they are playing a role in chemosensation. Now, important for this talk, one of the bacterial families that we identified is Enterobacteriaceae. There have been similar metagenomics studies of insects that have consistently identified Enterobacteriaceae. For example, you have Wigglesworthia. It's an obligate mutualist of tsetse flies, and they encode vitamin biosynthesis pathways that can supplement the tsetse's blood-specific diet. So much so that Wigglesworthia has to be present during larval maturation for the tsetse's immune system to develop and function properly during their adulthood. We also have Sedalis. That's a facultative commensal that's associated with stink bugs and with grain weevils. And Sedalis has been shown to impact insect vector competence and longevity. Now, for the purpose of our investigation, Enterobacteriaceae also includes the ubiquitous commensal E. coli. So we decided to first investigate E. coli's role within the mosquito because it's readily available, relatively harmless, and it's easy to culture and genetically modify. And what's more important is that not much is known about E. coli's role in insect biology, aside from that there are E. coli infections in the hemocoel of African malaria mosquitoes, and this can increase the mosquito's ability to fight off any subsequent E. coli infection due to a surge in hemocyte synthesis and nitric oxide synthase expression. But other than this, not much is known about whether E. coli functions as a behavioral manipulator in insects. So we investigated how microbes, including E. coli, within the insect can lead to changes in olfactory signaling and therefore the resultant olfactory behavior. And this could be as simple as changing olfactory neuron tuning or sensitivity, or it can involve an array of physiological changes to the insect which can alter their fitness. Luckily, many components of the insect olfactory system are broadly conserved, allowing us to utilize Drosophila melanogaster as a model organism for these harmful insect pests. Now, in these insects, the antennae and the maxillary palp act as their nose. If we take a closer look at the antenna, you can see that it is covered in these hair-like projections called sensilla, which are utilized to detect the odors. And these sensilla are distinguished by the types of receptors that they contain. Typically, these are olfactory receptors, ORs, or ionotropic receptors, IRs. And both these ORs and IRs sense volatile compounds, but they're distinguished in their ligand specificity. If we look even closer at a single sensillum, you can see that they are covered in pores. And these pores allow odorants to flow through and bind to these ORs or IRs on the olfactory neuron dendrites. And that movement through the sensilla is assisted or thought to be assisted by soluble proteins called odorant binding proteins, or OBPs for short. Important for this talk, the dozens of odorant binding receptors that are in each family do require a co-receptor gene in order to function. So for the ORs, this is Orco, and then for the IRs, you have IR 25A, 8A, and 76B. So these co-receptors are required for trafficking and signal processing. So it turns out that a significant factor that regulates olfactory function is the infection status of the insect. Matthew's team recently discovered that microbiota in general are modulating the olfactory neuron function. So you have here Drosophila larvae that are treated with antibiotic, and they exhibit this reduced chemotaxis response towards odorants compared to their infected controls. And this behavioral phenotype is partially rescued in larvae that are treated with probiotics. It appears that the microbes are specifically disrupting olfactory function by regulating expression of GABA and GABA receptors, all of which are known components of the insect's olfactory system. Wang's group also discovered that a bacterial symbiont of insects called Wolbachia has a significant impact on the olfactory response in Drosophila, this time in adults. So here we have flies that are infected with Wolbachia, and they're navigating towards a lure, and they're doing this much faster than uninfected controls. And that increase in that olfactory response appears to be caused by an increased expression in the olfactory receptor OR83B, otherwise known as ORCO, in the infected insect hosts. Now, in a more recent study that we conducted, we discovered that Wolbachia also triggers the expression of the odorant binding protein OBP6 in tsetse flies, as well as its homologue OBP28A, which is in Drosophila. When we deplete this OBP, what we result in is an altered olfactory response to a broad range of odorants, and I'm just showing you response to one octanol here. Now what you see is that the insect is getting a more robust and sustained response to that one octanol stimuli as compared to the control. So what we think is happening is that these OBP28A deletion lines, which are analogous to lines that don't have Wolbachia, they're not able to clear their noses. So once they receive a stimuli, they can't really can't perceive anything else within that environment, and therefore they become incapable of navigating towards stimuli in a complex olfactory environment. Okay, well, knowing this, we first wondered whether E. coli, which is one of our hits in that metagenomic study, could influence insect olfactory behavior. So what we did was we prepared exenic insects, we removed their eggshell, and we raised these insects on sterile food for 3 generations. Then we spiked that food with E. coli, raised them for 2 more generations to ensure integration of only that microbe. And then we test their olfactory response first using a trap assay. The trap assay is a primarily olfactory assay. And how this works is that you put flies into an agar plate, which has a little bit of agar just for moisture, and you present them with a choice between a trap that just has food or a trap that has food with some sort of a volatile, whether that be apple cider vinegar or ethanol or anything else that's attractive or repulsive. You put about 10 flies into this arena, give them 24 hours to make a choice. The way that the traps are designed, once they go in, they can't really come out. So within those 24 hours, you can record their preference index, which is the number of flies that go into the baited trap minus those that go into the control divided by the total number of flies. And what we expect to see is that in these exenic flies, we will observe an altered food-seeking behavior. And then if E. coli is influencing olfaction, when we reintroduce the E. coli, we should observe stronger food-seeking behavior. And we took this a step further, and we used lines that have deletions in that co-receptor IR25A for the IRs or in ORCO for the ORs. So what we're asking is, if E. coli is acting through a certain receptor class, then when the fly line that has the corresponding co-receptor deleted, when we reintroduce E. coli there, we shouldn't be able to rescue the olfactory signaling. Okay, so what we found, compared to the wild type, which has their microbiome intact, which has a very robust attraction for this apple cider vinegar, when we have exenic flies, which are in black, we either avoid now the attractant or we just don't go into either trap. So if you have a preference index of 0, that's like apathy for either trap. So what this is suggesting is that, yes, these exenic flies have defects in their olfactory coating and or have lost olfactory perception altogether. When we reintroduce E. coli, we increase that ability to navigate towards food in the wild-type Cantonese or in the ORCO deletion line, but not in the IR deletion line. So these results suggest that E. coli does influence insect olfaction. And it's happening through the IR family, not the ORs. And we've seen this to be true for the apple cider vinegar that I just showed you, and also for 10% ethanol. To confirm and extend these results, we then utilize the CAFE assay, which is also primarily a test of the olfactory system, but also includes gustatory response. So here, how it works is we have a 50 ml conical that we have punched 4 holes into the top, and we put capillaries in with a set amount of a feedant. Here we have either water or a gradient of sucrose solutions. We give the flies, we starve the flies, put them into this apparatus, give them 24 hours to drink from whichever capillaries they prefer, and then we come back and measure to get a volume of consumption from each of those experimental liquids. Okay, so from this assay, what we're seeing is that E. coli is also playing a role in gustation. Here in the axenic flies, again, in the black, you're seeing that they have this decreased attraction to any of the liquids that were presented to them. And then when adding back E. coli, we are able to rescue some of that appetitive behavior. And we see this for the Canton S flies as well as with the orchodeletion lines. But again, we don't really see that rescue in that feedant response in the IR deletion line. So it appears that E. coli is playing this role in gustation also through the IR family. And we might expect this because IRs have recently been mapped to gustatory sensilla as well as being co-expressed with gustatory receptors like IR76B. So now that we had a hint that E. coli is involved in gustation, we wanted to get temporal resolution. So we used this assay called the fly liquid food interaction counter, or the FLIC assay. This is specifically made for Drosophila feeding behavior, and this provides a precise and continuous quantification of the number and duration of interactions that a fly is having with food. And it complements those conventional methods of analysis, such as the CAFE assay and tracer dye approaches. But in this assay, we have the ability to do comprehensive long-term studies of these new and subtle aspects of feeding behavior, such as time-dependent changes in food preference or even individual levels of motivation and hunger. So how we set this up is we have these 2 channels and we flow a solution that's appetitive to them. In this case, we're doing 10% sucrose, so that all of these wells have a layer of liquid underneath them. Now, if I were to open the casing, you'll see that there's a metal ring around where the well of liquid food is. And when the fly comes and contacts the metal ring and then sticks out their proboscis to drink the liquid, they complete the circuit and the DFM can pick up that they have tried to feed. Then it continues to, calculate until the fly lets go of the liquid, and then it will decide whether it was just a taste or whether it was an actual feeding episode. Okay, and then what you'll get is these graphs which show, yes, they have fed, meaning that they have been in contact with that liquid for an extended period of time, or they were just tasting it. Okay, so from this analysis, let's look at the wild-type flies. We see that they, with their microbiome intact, really love to drink the sugary water. But when we go to the axenic line, we see that they aren't really tasting that sucrose solution anymore. But when we add back E. coli, we are able to rescue that feeding behavior. Okay, and again, in the wild-type fly and in the orchodeletion, but not in the IR deletion line. So we're seeing here E. coli is playing a role in appetite regulation through increasing exploration of food. And this actually results in them having fewer overall feeding events. But when they are feeding, they're feeding for a longer period of time. Okay, so it seems like when they're axenic, they don't have their microbiome intact, they're just trying out the foods. But when they have at least E. coli reinstated, then they have a very purposeful feeding behavior. Okay, and I don't know where that graph went, but they also have more rapid succession of their feeding bouts. Okay, here we go. So there's more rapid successions in their feeding as well. Okay, So indeed, in mammals, we have seen that intestinal infusion of E. coli stationary phase proteins suppresses food intake. It activates a noxious response that can result in long-term changes to their meal patterning. So to our knowledge, this is the first record of seeing E. coli affecting appetite in a similar way in insects. And this finding improves our understanding of insect nutritional ecology and illustrates mechanisms that insects are using to deal with the impacts of, let's say, global climate change or nutrient cycling and host availability. So thus far, we've looked at E. coli's function in insect adults. We have hints at E. coli's role in olfaction, gustation, and appetite. But this is not the only mobile stage for insects. Larvae also possess peripheral sense organs that can receive environmental information such as olfactory, gustatory, thermosensory, photosensory, or mechanosensory. The difference is that they're one large external organ that is coded by a mishmash of all of these receptors. It's not so much localized as they are when they are adults. Okay, so the other difference between larvae and adults is that adults primarily use their sense of olfaction to identify their targets. That's why we've been probing olfaction and gustation, which are more so long-range ways of finding their hosts. Larvae have to utilize a combinatorial code of their taste, smell, touch, and temperature to find their hosts or wherever they want to navigate, right? It's a little bit different for them just because of how they move. So with larvae, we can then assess whether or not E. coli or other microbes are altering these additional modalities beyond just chemosensation. So we're first curious about cold avoidance behavior. This is normally mediated by trip channels, which, yes, do interplay with the IR, specifically IR21A and IR25A. Drosophila larvae, they tend to have a weak tolerance to chilling. They can survive when approximately 50% of their water in their body has frozen. But they found that select symbiotic microbes like E. coli can produce these ice nucleating agents that can further moderate freezing in their hosts. So here we're wondering if E. coli can assist the host a little bit more by mediating this noxious cold perception. So the stereotypical behavior in Drosophila larvae towards this acute noxious cold is usually contracting and staying in place. They like to withdraw their head and then they completely contract the body. So we're grading that level of contraction here. Okay, so if we're looking at the control line, which has their microbiome intact, unfortunately we see there's no significant difference in that intensity of contraction, whether we have exenic or just E. coli added back. So it seems that E. coli is not really playing a role in mediating this cold nociception. Well, we were next curious about E. coli's role in insect heat sensitivity, which is mediated by a different trip channel, which is also mediated by IR25A. Now, insects' physiological activities depend upon their environmental temperature. So most insects are using heat sensing to locate food. With environmental increase in temperature, there are certain heat-tolerant microbes, again, like E. coli, that are enriched in the insect to help it to adapt to that warmer temperature. So again, here we're wondering, can E. coli further benefit the insect's fitness by mediating that heat sensation altogether and helping them to avoid it? Well, the stereotypical behavior in these wild-type flies with their microbiome intact is that they are moving faster. They're turning less often. They're more directed at getting away from a noxious heat. So we're grading, again, that movement intensity. Compared to the wild type with the microbiome intact, we do see that exenic larvae are moving a little bit less to get away from the intense heat. And once we reintroduce the E. coli, you can see that they regain a little bit of their rapid movement in response to a heat source. So this seems to suggest that E. coli is functioning to regulate thermosensing pathways, potentially through, again, an IR, like IR25A, to help facilitate escape from non-permissive temperatures. Because again, we get this rescue in the movement from the wild type and the orco deletion, but not so much in the IR deletion. Okay, so we then wondered if E. coli is playing a role in mechanosensation. This is again primarily mediated by TRP channels, but it's also mediated by IR93A, 21A, 40A, and 68A in Drosophila. Now, Drosophila larvae continuously explore their surrounding substrate during foraging and wandering. So they'll do head sweeps. If they notice that the substrate's a little too thick, they will try to move away from it. So we try to replicate that in the lab by getting an eyelash, putting it on the tip of a toothpick, and then poking the larvae at the head, the thorax, and the abdomen to see how they are responding. So far, not much is known about how microbes are playing a role in regulating this overall sensory process. So we're wondering, can E. coli assist the host by mediating mechanosensation? So here we're seeing that when we tap the larvae and the wild type with the microbiome intact, they will respond. They will try to move away. They'll typically do head sweeps or roll away, and we're grading that response. When we look at the exenic lines, we'll see that they just overreact. But then when we add back just E. coli, they have a more directed response. So if you tap them on the left side, they always turn to the right. If you tap them from behind, they try to squinch away faster. So their responses are more predictable when they have at least E. coli back versus when they're exenic, where they just respond randomly. So this is suggesting that E. coli is playing a role in tuning nociceptor output, but it doesn't seem like this is through any of the classes that we've interrogated, because we see the same thing with all of our deletion lines. Okay, so it doesn't seem like it's facilitating this primary sensation of the noxious mechanical stimuli. We next questioned whether E. coli can affect phototaxis behavior in larvae. Like most fly larvae, Drosophila larvae are negatively phototactic during most of their development. This is because they have a lot of pests in the environment that they're trying to hide from in the soil. So phototaxis is primarily mediated by photoreceptor neurons that express rhodopsins. However, there are some chemosensory receptors that have been found to play a role in this photosensing. So we just wondered, okay, does E. coli also function in that circuit to govern orientation behavior? So essentially what we're doing here is we have a plate that's half blacked out. We line the flies up in the median between light and dark, and we ask which way do they go after a period of 5 minutes, and we're live tracking this. And what we discovered is that normal larvae, they will try to go to the dark. Exenic larvae also will try to hide in the dark. But then when we add back just E. coli, for some reason we're getting larvae that are now preferring to go into the light. So we're seeing actually an attenuated preference for darkness. So maybe there's some other component here that we're missing, which would kind of mediate the response that we're seeing from just adding back E. coli. But of course, photosensory response is very complicated. It's segregating after the first synapses after the photoreceptor cells. So it's plausible that E. coli is acting upon some of those other pathways, let's say like the TRPL olfactory receptor, that can complement light sensing and then feed into this decision making. Okay, so now that we have evidence of E. coli being involved in at least olfaction and gustation in the adults, and then some other puzzling modalities in the larvae, we wondered if there was a general defect to the fly's physiology, particularly asking about cuticular hydrocarbons. So insect cuticular hydrocarbons, they help to prevent desiccation. They serve as chemical signaling that can help to mediate social interactions. Drosophila melanogaster's cuticular hydrocarbons have been studied extensively, but the environmental basis for individual variation in this composition is largely unknown. So here we're asking the question, does E. coli regulate cuticular hydrocarbon synthesis and its makeup in flies? So to do this, we take 5 male, 5 female flies, because we know there's disparity between the genders. We put them into hexane, let them incubate with vortexing, remove the flies, and then run the extract in the GCMS. And I'm just showing you the data from wild-type flies. What we see is that E. coli is affecting the composition of the insect's cuticle. And what we're seeing is compared to exenic, which is in black, when we add back just the E. coli in gray, we have a shift towards these shorter straight chain hydrocarbons. We also see an overexpression in 9-trichosine. And these compositional shifts did not alter the total cuticular hydrocarbon abundance, suggesting that the microbial influence is targeted to these biosynthetic pathways rather than global production. Prior work has shown that microbial colonization in general can alter cuticular hydrocarbon profiles, specifically by modulating enocyte transcriptional programs. And our findings are extending this now to E. coli, linking that bacterial presence to this pheromone signaling. Because 9-trichosine has a major role in mating dynamics, these microbial effects could reshape reproductive outcomes and potentially promote microbial transmission when you are in a more microbe-rich environment. And I didn't show you the data, but we did look at the IR deletion lines as well. And we basically saw the same trace for the hydrocarbons between the exenic and with E. coli reintroduced. So again, we think that there's a link to IR expression with the cuticular hydrocarbon synthesis. So today I've showed you that these insect-associated microbes are plentiful in non-gut tissues in insects. Across both larval and adult stages, the E. coli exerted IR25A dependent effects on thermosensation, tunneling, olfactory attraction, and cuticular hydrocarbon composition. They also modulated phototaxis and mechanosensation through some IR-independent pathways. This suggests that there's at least 2 mechanistic routes. We may see direct sensory modulation, where the E. coli is creating some metabolite that's acting as a ligand for IR25A containing receptor complexes, kind of like how you have bacterial acid detection in mosquitoes. Or we could be seeing a systemic modulation to the insect. Here, we could see that infection is triggering metabolic and immune changes that secondarily influence this sensory neuron responsiveness or other peripheral traits such as cuticular hydrocarbon synthesis. Nonetheless, our findings are adding to a growing body of evidence that microbes are remodeling their hosts' sensory landscapes. I already told you a bit about Wolbachia enhancing the olfactory receptor expression in Drosophila. We have so much evidence showing that gut bacteria are shaping this odor-driven feeding preference. And the fact that we're pinning something down to IR25A, which is a highly conserved co-receptor across insect orders, it suggests that this microbe-mediated modulation of IR pathways is a widespread phenomenon. So presently in my lab, we're conducting a screen of the KEO mutant collection, which is a systemic library of single gene knockout mutants of E. coli. We're subjecting flies that have received individual mutants. We're subjecting them to a simple Y maze to look for mutants that display a shift in valence. If we find any, these are potential genes that could be mediating E. coli's effect on insect olfaction and overall physiology. So that will be a first step at deciphering the pathways that are enabling these microbes to manipulate their insect hosts. We're also trying to identify things that are being secreted by the microbes, whether these are volatile compounds, quorum sensing molecules, maybe even toxins that are acting as signals or stressors to change gene expression in their insect hosts. We've also expanded beyond E. coli. Now we're interrogating the regulatory capacity of other ecologically relevant bacteria and fungi. For example, where we have some evidence from Pseudomonas and from Serratia that are also linked to improvements in vector competence and host immunity in Drosophila, and then we're moving into mosquitoes hopefully soon. We've also seen some implications for Pseudomonas and Serratia in insecticidal activity. And overall in regulating, again, that IR pathway, similar to the story that we've shown with E. coli. So it suggests that all these bacteria are doing more than just altering their host immune system and survival broadly, but actually altering components that are inherent to vector competence through shifts in host seeking, oviposition, and feeding behavior. Another thing that we're studying, actually Hunter's spearheading that, we're studying competition amongst these microbes to understand which species are dominating within their hosts and how that winner of all of the competition can directly affect whether the insect becomes permissive or resistant to viruses, parasites, or maybe even fungal invasion. All said, our investigations should reveal the acquisition routes, the persistence, and the roles of these microbes on host physiology. In doing so, our innovations promote sustainability by advising strategies for using predictive analytics so we can predict areas that are likely to experience outbreaks based on the microbiome shifts and insect behavioral patterning. We could use insects as bioindicators of pollutants like heavy metals and industrial chemicals in our environment, utilize microbes to reduce vector competence and survivorship, use microbial interference to reduce pest survival, maybe even make crops less attractive to their pests. We could improve the quality and safety of insect-based proteins for human and animal consumption, and even optimize insect-driven food waste conversion. With all that being said, my team is in its infancy. I started here technically 2.5 years ago as a presidential postdoc, where I met my first students who have helped me get a lot of the data I've shown you today. But I started this January as an assistant professor in the department, and now I have even more students who are working on all the projects I mentioned in the latter half. But we would like to invite more people to join our team. So if you're interested in what we do, definitely reach out. I'm part of the microbial biology and the molecular biosciences programs for graduate students. And thank my funding sources and my collaborators. And I'll take any questions.